Okay, guys. Hopefully we're recording. We're recording. We're recording. There's just going to be two different files. I was about to throw something. Sorry. I'm under a lot of stuff. Okay. So we were just talking about actin and myosin, and remember I said it's a superfamily, which means there's many different proteins in this family, but they have a similar structure, right? They contain a conserved N-terminal motor domain and a divergent C-terminal domain. Why? Why do they have a conserved N-terminal motor domain and a divergent C-terminal domain? What does that mean? Yeah.
It means it will bind to the same filament, so actin and microtubule, but it will bind to different substances, so it can transport different things.
Right, so the motor domain is conserved, so it will bind to the same thing. It has the same structure. The structure is what dictates its interaction with other things, right? So it's going to bind to actin. They all have the same motor domain. They all bind to actin, but they have different C-terminal domains. Well, that's the part that binds to the cargo, and so they're going to have different functions. So, for example, here's your myosin 2 with this nice long coil. And so its C-terminal domain actually, it doesn't really have a cargo, does it? It really binds to other myosins and forms a bipolar-thick filament. But then there are these smaller ones that are going to bind to cargo and walk along actin. So different C-termini because they have different cargo. Conserved motor domain because they all walk along actin. So here are some interesting myosin facts. They're not only present in muscle. So myosin, don't only think muscle, right? Not only muscle cells. Myosin's also found in protozoan cells and plants, but mostly in eukaryotic cells. 9 out of the 40 human myosins are expressed in hair cells of the inner ear. Fun fact. Mutations in 5 of these myosins are known to cause hereditary deafness. Who knew? I did not know this before teaching this class, that hereditary deafness is associated or caused in part, obviously not all, with mutations in myosin. I thought that's cool. It's important for the construction and function of stereocilia. That's those hairs in your ear. All but one of these myosins move towards the plus end of actin. So as a rule, you can kind of think plus end. Myosin walks to the plus end of actin, except for one. And I will never test you on the exception. Right? That's really silly. So some functions of different myosins. Myosin 1 binds actin and the plasma membrane, and it's involved in cell surface protrusions. Myosin 2 is involved in contractile activity in muscle and non-muscle cells. It's required for cytokinesis and cell migration. Myosin 5 is involved in vesicle and organelle transport. But most of these myosins, their function is unknown. So actually, if you're kind of trying to decide on a research project in a lab that studies myosin, you can choose from a whole bunch of them that have no known function and just try to figure out what they do. Assuming your boss has money for that. Okay, so let's move on to kinesins. So kinesins are microtubule motor proteins, many of which are involved in mitotic and meiotic spindle formation. Again, conserved motor domains make sense, right? They all walk along microtubules. They have a similar structure as myosin, that kind of globular head in a middle coil region. And then they also have a kind of a tail domain, which is going to interact with cargo. Most walk to the plus end, except one. So you can be safe to think kinesin, plus end. Kinesin 14. for whatever reason. Doesn't walk, it walks to the minus end. Dyneins are motor proteins that walk to the minus end of microtubules. And they're the largest and fastest, yes,
On the last slide,
yes.
kinesin 13 is right there, so I didn't know if it was a motor protein.
Kinesin 13 is right there. Well, let's do that. Let's do the experimentation. I mean, it is kinesin 13, so it has structure, right? It does look like it has head domains, but then it's got this. What are these moving? So notice the structure is actually quite different, right? That's a very different structure than these guys. Well, then this one. This one's quite different too. So maybe it is a motor protein, and maybe the way it causes catastrophe has something to do with how it's interacting with those alpha beta tubulin dimers. I have no idea. So now I really want to know the mechanism. So this is why sometimes more detail is better, because you can understand things much more deeply. Yeah, good eye. Good eye. All of you. You all have good eye. Everyone. You're all special. Okay. Okay, thank you. Dyneins are motor proteins that walk to the minus end of microtubules. They're the largest and fastest molecular motors. So they're different kinds. Again, these are all families of proteins. There's cytoplasmic dynein, there's ciliary dynein, also called exonemal dynein, which drives the beating of cilia and flagella. Cytoplasmic dynein is important for vesicle trafficking and localization of the Golgi near the cell center, where it should be. So myosin, so you might have noticed myosin and kinesin have similar structures. They share a structural core. So that ATP binding domain in both of them is quite similar, suggesting they have a common evolutionary origin. So this is myosin here, and this is kinesin over here, and it's showing that you've got a similar ATP binding region here. That's interesting because they have actually different tracks, right? And so actually the actin binding site is very different from the microtubule binding site. That makes sense. Also, these proteins are such structurally similar to the GTP binding domain in RAS. Interesting. What do we know about RAS function? What does RAS do? Who did the brain dump? No brain dumping allowed in this class. What is it? It's a molecular switch. What can you tell me about RAS in terms of its GTP binding domain?
Does it orient where the vesicles go?
No, that's RAS.
It causes a conformational change that allows, I guess, things to bind to.
Okay, so going very, very basic, when we talk about GTP binding proteins, we are often thinking about the fact that GTP binds hydrolyzed conformational change that causes binding to something or not. And so that, I think, is what the significance is of the fact that the ATP binding domain and GTP binding domain are similar in myosin, kinesin, and RAS, which is like such a different protein. We're just talking about similarity with one domain, which is that nucleotide binding domain. And with all of these, when you have nucleotide hydrolysis, you get a conformational change, right? You guys believe me when I say that? Right? Remember we talked about how the size of ATP and the size of ADP are kind of different? And so that's going to affect the shape of the protein and its function. So in myosin and kinesin, that's kind of affecting the swinging out of the head domains and that movement of those motor proteins. In RAS, it's a conformational change that is turning off the protein. So this was RAS was the molecular switch that is involved in signaling, right? It spreads the signal. Remember, there was phosphorylation, recruitment of the RAS gas, which then turns on RAS, which is the plasma membrane right next to the receptor. Maybe. So that when you start to see similar domains, you can start to think to yourself, well, how are these proteins similar? So myosin uses ATP hydrolysis induced conformational chains to generate force. Right? So again, you've got ATP. ATP is being hydrolyzed, change in conformation, and that's generating force to move forward along the actin filament. And he uses this rocking lever arm. And so you've got your myosin head, and then there's this light chain here, which is this. It's basically acting as a lever arm. Who's taken physics? You guys learn about lever arms? Maybe. Who said yes? Unfortunately, is that what I heard? Physics is awesome. The problem is there's no physics for biologists. There was talk about making a physics for biologists class, and then you can start thinking about the physics, the biophysics, right? And then you'd be like, oh, this is so cool. Like kind of like chemistry becomes very, very cool once you get to biochemistry, right? Or not, or it's just all horrible. I really want to digress right now, but I'm going to keep moving forward. Okay. So here you see there's no ATP attached. And so it's very tightly bound to this actin subunit right here. But when ATP binds, it changes the conformation of the head, right? So now it can't bind to actin anymore. And so it's kind of just hanging there. And so then you're going to get ATP hydrolysis. It's still bound to that phosphate group. So that changes its conformation again. And kind of, you can see here you've got these two little blue things facing to the left. And here it's going to rock the other way. So that lever arm is going to go like this. Right? Lever arm. Oh my God. I know, guys. You're doing great. Okay. And then when you have the release of that phosphate, you get yet another conformational change. And so then it's going to bind. Then it's going to bind to the next actin. So it's starting over here, and it's going to that actin subunit. Right? So then it's going to release the ADP, but then again be binding tightly to the new actin. And so that's how it's moving. It's really all about ATP and how it affects the shape of, or the orientation of this lever arm that's associated with myosin head. Okay? And it's basically going like this. I have a movie, but in order to not crash my computer, we're going to play it over here. Hopefully. Or maybe we don't have a movie. Huh? I didn't test this link this morning. That's my fault. Okay. That makes me mad. See, it says there's a link. See it? It's right there. Oh, wait. Yeah. It's a YouTube video. Okay. Let's see if I can find it. It's a fun one. It's going to correct it for me. Let's see. Okay. Well, let's do this one just for fun, because movies are fun. Even though we're Really? Well, Avi's silver. Oh my God, it's the I thought it was just gonna be movement, but no. Okay, you guys could. Okay, so I know you love it. You guys could look that one up yourself. It could have been way worse. Imagine what I could have clicked on. Okay. Okay. So apparently someone made a song about this. I'll try to find the video that I was trying to show you guys and pass it along. Okay, so let's move on with Kinesin. I know that was fun, guys. I'm just waiting though for the day that I open up something horribly inappropriate and get fired. That was inappropriate in the sense that I didn't really plan on it, but it was not to get fired. Okay. So Kinesin also has the same walking motion, and it walks along the microtubule. Again, you've got this ATP binding kind of driven force, right? In this case, though, when you have ADP bound, the head is very loosely bound, and so it's kind of a different mechanism to myosin. I'm never going to ask you to memorize a mechanism of myosin or a motor protein's movement. Understand the basic concept that they require ATP. ATP changes the conformation of the protein, and in this case, the big part to pay attention to is this linker protein here. And so the conformation of that linker protein is what changes when you have ATP binding and then ATP hydrolysis. So when you go from ADP to ATP, you're driving forward motion of this rear motor domain. So you're watching this and you're going to go from here to here. So I have a movie here too. Let's see if this one works. So it's the opposite. It's the opposite of myosin, where ATP. Let me get back to it in a sec. So it has to do with loose and tight binding, basically, and then the conformational change. Okay. So here's kinesin. Is it going to work? Let's see.
The motor protein kinesin is a dimer with two identical motor heads.
Let's see.
Each head consists of a catalytic core and a neck member. In the cell, kinesins pull organelles along microtubules.
Let's start again.
The motor protein kinesin is a dimer with two identical motor heads. Each head consists of a catalytic core and a neck linker.
There's no linker.
Kinesins pull organelles along microtubule tracks. The organelle attaches to the other end of the long coil that holds the two motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the two kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ADP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes the ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats.
That's cool.
In this way, kinesin dimers move processively, step by step, along the microtubule.
think they really enjoy their jobs. So it's all about these linkers right here and that conformational change. Okay. Oops, no, that's not what I wanted to do. Okay. Okay, so then you have diamonds. And so diamonds are totally different structure, you'll notice. They've got a stalk that attaches their head. So, and then it's this kind of round structure with the where the ATP combined, and then it has the tail that binds to either cargo or another microtubule. So this movement is quite different. And I have a movie for that too, and then I'm going to let you go after that, but at least I can have all of the motor proteins covered. Maybe. Okay, guys, you guys will all figure it out. I'll send it out to you. Okay, that's what the exam is going up to. That is it.